with that, we have about some time to talk about a little bit more detail about genomic database and phenotyping. And Caren, you're going to be first because if that's okay with you.
Yeah, are we still aiming for 12:30 lunch?
Ish, yeah. I mean, we have an entire hour to eat lunch, so yeah.
I'm gonna try to condense what I say so that there's more time.
Sure. Leave it up to you. But people might have questions.
Okay. Oh, I'm gonna skip some. Okay. Okay.
Don't feel pressure, okay?
No, I feel pressure. Okay, so I'd like to give a brief update on the database. Again, we sequenced those 13 cultivars. And this is sort of an example of an output carried out by a bioinformatics grad student. I could spend time talking about this, but I'm not sure. Should I spend time talking about what all these things are? Or does that?
I don't think so.
Right, I can skip that, so, but what does the database allow us to do? We can, browse the sequences of any gene of interest. Again, we're starting with the genes in this case, and compare and analyze those gene sequences. And if there's a gene that is known to have a certain function, we can compare and analyze that gene in different cultivars and then see if, if the cultivar that I'm interested in has a defect in a particular gene. Using the sequence, we can design DNA markers for breeding. And we can also design, using the sequence, we can design guide RNAs for gene editing by CRISPR. And in my lab, we're able to transform strawberry fairly easily, but we're only using the diploid strawberry. So I don't know about octoploid strawberry. Does anyone do CRISPR in octoploids?
We do something. I mean, there's some labs that do.
Yeah, okay, so that could be where how the genome sequence could help. I was going to go through this database, but I don't think it's of so much relevance. I was going to show you how to use it. Basically, you have to know something about the gene of interest. You put in a search term, and then here's a search term that's an actual gene ID, and then you can view that sequence. Here's an example. It just looks like a bunch of dots because we're too zoomed out. We have to zoom back in to see the sequence here. Here's an exon, intron, exon of that gene that we search in the database. But looking at one gene is not just that one sequence isn't what the power is, it's the comparative power. So you can open this track selector, you're selecting other genomes to look at. So the genome data of the reference is Royal Royce, and we can click on any of these 12 other varieties. So let's look at Camarosa and Chandler for that gene that we entered. And now we get, again, this is the sequence, but we're too zoomed out. This is the gene structure. Exon, intron, exon. Here's Camarosa. These are reads from the sequence data, and below that is Chandler. We can zoom in more. Okay, so now we can start to see the DNA sequence. The 2 strands of the double-stranded DNA are in these colors, and then on top is a translation of amino acids in 3 different reading frames going in one direction. Going in the other direction, you have 3 reading frames. The red means there's a stop codon. And so now we can see a little bit of Camarosa and Chandler here, and the variants are marked. So are you understanding? These are, the gray means the sequence is matching. And then when there's something that doesn't match, it's marked by, and this is going to be in a genome browser for anyone to access. So it's supposed to be user friendly. So I'll just say that these are insertions, and it tells you the number of nucleotides that are inserted. There's deletions where you have a dark gray, missing sequences, and also single nucleotide polymorphisms. So we can use that to identify variants. So it's pretty easy. So if we have gene of interest, we go in and we look at the cultivars of interest to find the variants. What's a little harder is matching it to traits that have been seen. So I want to just give a quick example of the GA20OX4 as our best example currently. This is involved in biosynthesis of gibberellic acid, and we saw that there is a insertion in Fronteris. No other cultivars have it. So there's a 1 base insertion, so there's an A, and that's going to throw off the reading frame, so the protein's not going to be made properly. Monterey and all the other cultivars are normal in all subgenomes. But Monterey was examined, so we can look at the phenotype of Monterey versus Fronteras, because the runnering, we believe GA is involved in producing runners. So we can expect from this that Fronteras would have a little less gibberellic acid and so might have less runnering. And that is in fact what Ibrahim found. And I think he'll be talking about more detailed phenotyping after me. So then we went on to make a marker because we had correlation, and this is the only marker that we have so far. And here we have 3 cultivars that have the wild type sequence. As I said, they're all 12 have the wild type, but we just looked at these 3 and we did 3 replicates, and that is seen in this melting curve. What this is an increase in the temperature. So as we slowly increase the temperature, we're looking at the melting of the double-stranded PCR product. And as it melts, it will melt differentially for the Fronteras because of that one base difference. And the wild types are all going the same way, the 3 replicates per wild type cultivar. And then the 3 Fronteras are all melting differently. So this is a very clear difference. So if you were using this in breeding, you can take the seedling, look at it, and using this assay, and then we know what might happen later in the runner phenotype. So, so just to review again, I showed this, we start with the gene in that forward way using the database I just mentioned briefly. And then we have to identify the cultivars that carry those high impact variants. But then the kind of bottleneck we discovered is determining whether those identified cultivars actually display the traits that we predict, because we have a small list of genes. Even though we looked at 90 genes, we only found variants in maybe 15 known genes. So that's 15 traits. And again, I don't think it's going to be a one-to-one. You have one gene, and then you're always going to see that phenotype. That's kind of rare. It's an interaction of genes with the environment. As you all know, changing the environment is going to change how the plants grow. So I wanted to now talk about this opposite method, which was not initially proposed, but Ray came up with this, where we start with the traits or the cultivars, and then identify all the variants in those cultivars. And if the variants are there in genes, we don't have to know anything about the genes, and that's the power of that. So I'm going to give you some information about that. That's probably what we want to focus on in addition to the original direction, but we may be running out a little bit of what we can do there. So UA created just an Excel spreadsheet of any cultivar. So there's actually, this one only has 12. We didn't have the 13th yet. These are the 12 cultivars. And if there is a high impact indel, this is right now, we're only talking about indels. If there's a high impact indel in any one of the cultivars, it's in the list. So this gave us a list of over 9,000 genes down here. So the list starts off, so it's not 9,200 genes or more. We're just going in order. So
I have a
yeah.
dumb question. Sorry, I have a dumb question.
Sure.
What's a high impact?
Oh yeah,
There.
high impact means it's going to change the gene product, the protein.
Okay,
So it's in the coding sequence like a stop codon, but we're not doing SNPs, we're doing insertions and deletions. So this spreadsheet has the genes according to the order of chromosomes, subgenome A, and then B. So for the 7 chromosomes in subgenome A, then 7 in B, 7 in C, 7 in D. There's the best hit in Vesca for Garrya vesca. We have the gene name there because that's been more annotated. We have the gene name, which can come from Vesca or it can come from Arabidopsis. There's description of the gene function if known. And then there's a gene ontology term. And then the 0 or the 1 is telling us if there's a high impact indel. So albion has 0 for this gene, but we see 1 in Cabrillo, Chandler, Finn, Fronteras, etc. So 1 means there is a hit. So this database can be sorted. So we can find high impact indels that are unique to just one cultivar, like Fronteras seems to be somewhat odd, or albion doesn't seem to root, someone said earlier today. So if we can find what genes are specific to albion, what genes are defective, whereas all the other ones have zeros, but albion has a 1, we can look at that list and see if anything has to do with rooting. Right, so it's backwards. We don't have to start with genes that are known to be in rooting. We will discover the genes. So an example that Yue just did just randomly, because we don't know so much about the phenotypes. We asked, can you find all the genes with high impact indels? He just randomly chose albion and frontaris. And not in Monterey. So you can do these nice comparisons. If you never see a trait in Monterey, but your trait of interest is in albion and frontaris, you can sort this list. So here albion has a 1, meaning there is a high impact indel in all of these genes. And it's also, there's a 1 in frontaris, but all of these genes have a 0 in Monterey. So hopefully you can see the power of this because we can do all different combinations of analysis. How do we do the sorting? So at this last column, so all of the cultivars have a letter associated with it. And in column P, it says group A, and then there's column for group B. So you can choose the cultivars that you want to compare. So K is Monterey, D and J are Albion and Fronteras. And then what we do is just look at the output here, A versus B, group A versus group B. We can see 3 different outputs. Same means they all are the same. For indels, either they all have one or they don't. Or they're different values detected within the group, meaning between Albion and Fronteras, there is a difference. Okay, you get it? So there's 3 outputs. If then we can sort, because what we really want are genes that are the same. The indels are the same in D and J, not in K. So the comparison result is here, but we can sort, filter basically for different. Or you can choose same if you want or whatever. So if we choose different, now we get all the ones that are different. And so these are the genes that satisfy our query. Query being same situation in D and J, but different from K. So you see how it works. So to me, this is very powerful. So we're excited by it because you can do any kind of combination of search. You can compare. I should have tried it, but it didn't have time. So like each one compared to the rest of the 11, right? Each one, what makes it unique? The set that comes out of it would be what's unique to that cultivar compared to all the others. It could be a long list because when we did this, we got 982 genes. That's a lot. So, but we did do this with the comparison with Fronteras Monterey, and we do see the GA20OX.
Well, and you're looking at every subgenome separately, right?
Yeah, every subgenome separately. So this is the huge, yeah. And this is only indels, not the SNPs,
Yeah.
which we will develop next. So that's a lot of genes, but I can show you some more examples in a minute. So we also can connect the indel, the 1 and the 0, to what the actual sequence is. So we have another sheet database of all the indels. And so the reference sequence is quite long here, but the alternate sequence is a deletion, so you only have a single nucleotide. This gene starts off with a T in the reference, but we have an insertion. So this shows us the actual sequence if we want to design markers or just know what's going on. And some of the genes are listed more than once. So like 222 is twice because 2 different positions of that gene have a change. So this is detailed information if we want it for sequencing or whatever, or not sequencing, designing markers. So let's do another example. Since I don't know too much about the phenotypes, we just randomly chose some. So we can expand instead of doing just one versus a group, we can take a group and ask if we make group A, these 5, and group B, these 4, we can ask, is there anything in common in these groups, this group, among these guys, or among these guys, either way, right? So we did that, and we only got 8 genes, but that's interesting. So these 8 genes happen to be all shared. These are genes with indels. They're shared, they're in common with these guys. And there's nothing in common, no indels in common with group A cultivars. We can look at those and they do have annotations. And they all are clustered. If you look at the location, they're all on a section of a subgenome 3C. which is kind of interesting. So something happened in that section that got carried along. So that was pulled out randomly. The only phenotype I really know about when we sequence the genomes is short day versus day neutral. We'll do that. So we did that one because I know that's a phenotype. So I took all the ones that I know, short day, compared that to a group of day neutral. We only got 2. So these 2 are indels in common in the short day group. So all of those share 2 indels. It would be interesting to know what these genes are. One of them doesn't really have any information. We tried to blast it. We don't know what it has anything to do with. So there's a lot to play around with here that might reveal some genes. And so we're excited about doing that. So one possibility is, see, there's kind of different possibilities. If we have information from you about cultivars that look the same or doing something, then we can put it in here or make the sheet available. And you just play, I mean, it's very easy to just fill out and see if there's a gene list. And then we can take that gene list, because that's sort of gene discovery, and we can look further into that. Another way is if we play around with this and we see something like, oh, hey, these guys have something, well, let's go to the previous one. These guys have something in common. Do you see anything in your work that would make them, have the same trait? Okay, so that's my summary. So we have a database that carry of all genes that have a high impact indel in at least one of the varieties. And the big advantage here is that the user now is just entering cultivar names, right? That's way easier than entering gene names. So you enter the cultivar names and to get a gene list, whether you know those genes or not, what they are. And no prior knowledge of the genes is required. And the user can then rapidly, within seconds really, you just push the filter, you can identify all the genes that carry an indel that's unique to a variety or unique to a group of varieties. And such variant genes and their go terms have the potential to be responsible for the particular traits of interest. And so I think this tool, this going in this direction, allows us to discover genes and pathways that we didn't know about prior in the cultivars of interest that you're interested in, as long as we have the sequence for it. And we are now expanding to look at SNPs, and we just need to play around with this a lot. Okay.
Cool.
And that work, the prior database, these 3 did a great job. And using that database, UA created the indel database that we're now looking at. Thanks.
Thank you. You have any questions?
Well, how much, is this the only collaborative database? Is everybody collaborating across different programs, or? Cause I know there's at least 2 proprietary gene
We
editing.
don't have proprietary stuff.
I was sort of saying, so
Yeah.
I think it's really valuable, but I'm just curious. Like there's these other databases, right?
Right.
But are they the same, different cultivars? Or proprietary cultivars?
It's both, probably,
Yeah.
they're probably benchmarking themselves against public ones.
So yeah, I'm pretty sure somebody like did come up with cardios before that, but it's not, this is really.
Just real quick on the Fronteras deal, so your prediction is that Fronteras will be harder to propagate at the nursery level? Or is that is that the implication of that?
Yeah, some of it
Yeah,
is.
But it's a good runner. I mean, it's not the best
It's not
runner,
the best
No,
one.
it's
but it's a moderately easy nursery propagation plan
under certain conditions,
with
maybe.
cold conditions.
Yeah,
So.
Yeah.
That's a factor that makes things really
Right,
complicated.
right.
Yeah.
because in the system, in the hanging garden system, it's not the worst runner. I mean, we have a lot of the everbearers are worse in that, like, in terms of running, but it's more the short days, it's not that great.
But it works, so that prediction makes perfect sense outside of the environment.
Yeah. Great. All right, so you all think you're in the south, but it's actually pretty cold in there. Last talk, we will talk now, we work with Caren in tandem on the phenotyping part, and we share those data. We just finished the phenotyping project, so this is fairly new data. And we look at 11 out of 13 in a repeated study in hanging garden. This was a lot of work.